Hi, I'm Joanne Corey, and the work described in a paper by Meng Cheng and others in this week's Cell began while Meng was a postdoc in my lab at the Salk Institute. We've been interested for many years in how plants respond to light in their environment. And because they're photosynthetic, plants have evolved a number of different photoreceptors and look to light for many aspects of information. So for instance, plants use light to tell them where they are, what time of year it is, and the time of day, etc. So plants do this by a series of photoreceptors which are defined by the colors of light they absorb. And these photoreceptors, there's at least 10 of them in Arabidopsis, and the subject of our paper is one of these red-absorbing photoreceptors called phytochrome B.
Welcome to Department of Biology at Duke University and Meng Cheng. As Joanne mentioned, that this project was initiated when I was a postdoc in the Corey lab at the Salk Institute. We continue to work on this project after I moved to Duke. My first two postdocs, Raffaello Gaval and Mei-Na Li, are co-authors of this paper. In this study, we focused on one of the earliest light responses, the formation of phytochrome nuclear bodies and its role in phytochrome signaling. The inactive phytochromes are mostly localized in the cytoplasm. When photoactivated, they rapidly accumulate in the nucleus, where phytochrome interact directly with a group of BHLH transcription factors that are called PIFs. Together, they co-localize to subnuclear foci that are called phytochrome nuclear bodies. Light also triggers the degradation of phytochrome A, or Phy-A, and the PIFs. The significance of Phy-A degradation is unknown. Because most PIFs are negative regulators for phytochrome responses, the current view is that phytochromes regulate gene expression by mediating PIF degradation. However, how PIFs are degraded and whether phytochrome nuclear bodies plays a role in Phy-A and PIF degradation is still unclear. In this paper, we show the first genetic evidence linking phytochrome nuclear bodies to protein degradation. Using a forward genetic screen to isolate mutants with abnormal morphologies of Phy-B-GFP nuclear bodies, we identified a novel phytochrome signaling component, Hemera, that is also required for phytochrome nuclear body localization. This screen also brought forward a couple of surprising discoveries. The first surprise is Phy-A and PIF degradation are also impaired in Hemera mutants. These results directly linked phytochrome nuclear bodies to Phy-A and PIF degradation. Although Hemera encodes a protein without similarities to known protein classes, its predicted secondary structure shows significant similarity to HHR23A, the human ortholog of the yeast protein RAD23. Which is a multi-ubiquitin receptor involved in delivering multi-ubiquitinated proteins to the proteasome for degradation. We show that Hemera functionally rescue RAD23 mutants in yeast, suggesting that Hemera could act like RAD23 in Phy-A and PIF degradation. The second surprise comes from the subcellular localization studies of Hemera. In a previous study, Hemera was identified as part of the chloroplast transcriptionally active chromosome complex, which contains plastid RNA polymerases and is required for the expression of chloroplast-encoded photosynthetic genes. Here we confirm their observation. More importantly, we show that Hemera is actually dual-localized both in the nucleus and the chloroplast. This might explain the phenotype of the Hemera mutants, which is both tall and albino in the light, and also leads to the hypothesis that Hemera could be involved in the coordination of both nuclear and the chloroplast gene expression when plants first encounter light.